Namaste students, Khush or Vidya. I JV and Dr. Chandrakala, Assistant Professor Jyoti Vidyapeeth, Women's University, Jaipur. In today's class, we are going to discuss about the reproductive structures and the male cones of the gametophytes in pinus. Okay. So as we have already studied about the pinus plant morphology, so today we will discuss about the reproductive structure. Okay. So the tree are monoecious. Here the male cone and the gametophyte, we can find the tree are monoecious. So the trees are monoecious, but the male and the female cones are both on the separate branches. Okay. The male cone arises from the lower end and the female cone on the upper branches. The male cones which replace the draft shoots occur in the cluster. Okay. The number of the male cones in the clusters varies from 1 to 40 or 140, depend upon the 15 to 140, depend upon the final species. Okay. So each male here consists of central axis, or here the central axis and which the microsporophylls are spirally arranged. Okay. Each microsporophyll bears two sporangia on the lower abaxial surface. Okay. So here we will see them. Okay. You can see here in this diagram the pinus, the shoot bearing cluster of the male cone. Okay. So here the first one is the shoot bearing cluster. Okay. Then you can see in the early stages, okay, in the early stages of the male cone, the changes taking place. Okay. So third is the lateral view of the microphyll. Okay. And the microphyll bearing two sporangia on the abaxial side. Okay. So microsporangia bearing in the abaxial side, and this is the LS of the male cone. Okay. These are the microsporangiums, and these are the microphylls, and here is the resins are deposited in the inside the cone. Okay. So next is the microsporangium. So here the microsporangium is as you are already familiar with the word microsporangium and with the structure or the male cone. In the male cone, the pinus, you can see here the developmental change lead to the formation of the male gametophyte. Okay. One or more hypodermal cells are differentiated and archisporangial cells which divide repeatedly into forming the archisporangium tissues. Okay. So here you can see the LS of the LS of the young microsporangium to show the hypodermal archiporal cells. Okay. And then here these are the archiporal cells deposited, and these are the epidermal cells. These are the pollen grains present here, and this septum which help in nourishing the tissues. Okay. So the one or more microsporangium. So one or more the hypodermal cells are differentiated into the archiporal cells, and which divides repeatedly into the archiporal tissues. Okay. So the peripheral cells in the archiporal divisions or the pericyclic to give rise to a primary parietal layer of the primary sporangium cells. Okay. The former cells divides and form the periclinically or anticlinically forming the remaining 2 to 3 layers are middle layer and which degenerate on the maturation of the sporangia and the primary sporangial cells on the other hand, okay, divided in the form of the plane or producing a mass of the sporangial tissues which last or the last cells to generate generations and forming the microspores mother cell stage. Okay. So the epidermal cells in the last stage or the later stage develops the fibrous thickening except for the few cells. In the row which forms or marks the resize, the development of the tapetum and the sporangium cells is stimulated or by the gymnosperm. Thank you. This session is powered by 2.0 Jyoti Vidyapeeth Women's University, Jaipur. If you have any queries, please write down in the comment box. We'll resolve in the